thank you very much, Helen. It's a great privilege to be here. I'd just like to start with the medical definition of an expert. An expert is someone who comes from somewhere else and brings slides. So I'm feeling a little bit naked today because I don't have any slides. But what I'd like to do is discuss several interrelated things. Firstly, why your eyes are so special and how some of the most innovative new medical breakthroughs are relevant to eye diseases. Secondly, some of the challenges we face in terms of macular degeneration. And thirdly, talk about the research that my group and others are doing to combat what's really an epidemic of blindness. And so we're going to talk about genes, gene therapy, stem cells, telescopes, artificial retinal implants. So there's a lot to cover. But firstly, just to say, our eyes are a miracle of evolution that even today modern cameras can't emulate. They function in the brightest sunlight and yet can also detect a single photon of light from a distant star. And that dynamic range is not found as yet in any human-engineered device. As your eyes spin around to change gaze, they don't wobble, you don't get seasick, they fixate automatically on what you're looking at, and that's also a remarkable piece of engineering. And have you ever thought that when you blink, you don't notice that your vision's temporarily blocked? Your brain's very clever at filling in that gap. So as we've heard, and is relevant to thinking about how we're going to cure macular degeneration, it's useful to think of how the eye works. So light comes in through the front of the eye, through the focusing mechanism of the cornea and the lens, and is focused on this tissue at the back of the eye, the retina. And then if we look at the anatomy of the retina, it's a bit like a sandwich. At the very bottom, you have these choroidal blood vessels that are very vascular, and given the chance, they grow up like weeds coming through cracks in the pavement and form these unhealthy blood vessels within the retina, and cause the scar tissue that the Lucentis injections and so on aim to avoid. And the choroid has an intervening membrane called Bruch's membrane, which is the sort of the wall that stops these blood vessels from coming forward. Then you have this layer of cells called the retinal pigment epithelium that provides nourishment to the cells that actually sense light, and that's the photoreceptors. And interestingly, our eyes wired back to front. Light has to get through the overlying wiring in the retina. There's a group of cells called ganglion cells, bipolar cells, and it goes through these cells and then strikes the photoreceptors and generates the electrical signal. In other animals, those cells are wired the other way around, so the photoreceptors are more at the front of the retina rather than the back. And we're unusual compared to many other animals. We have an area where that wiring, the ganglion cells and bipolar cells get out of the way so the light just strikes the photoreceptors, and that's called the macula. And that's a tiny bit of tissue, less than 0.5 millimeter in diameter, which looks after all of our central vision. And that's both a blessing and a curse. It's a blessing because it gives us wonderful sharp vision, but it's a curse because if there's any damage to that tiny area, it has a profound effect on your vision. So that's why you can, with wet macular degeneration, if it affects the macula, you can notice a dramatic loss of vision, sometimes even overnight. But it's an advantage in that it's a tiny bit of tissue if we're thinking about trying to replace it or cure it. And then we have another theoretical advantage, is that there's, from photoreceptors to brain, there are just 3 connections. There's a photoreceptor, bipolar cells, and then the ganglion cell goes all the way back through the optic nerve to your, and then connects with other neurons in the brain to give us our vision. So if we can make a connection to those ganglion cells, even if the photoreceptors are damaged, then potentially we could see again. So the challenge we face with macular degeneration is that it's a huge problem. There are 26,000 people in the UK registered as blind or partially sighted every year. So that works out at about 500 people per week who are registered as visually impaired. And not surprisingly, there are some famous people affected with macular degeneration. So I don't know if anybody's seen Last of the Summer Wine, but there are 3 individuals in that program, and one of them, Peter Salas, is affected with macular degeneration. So that age group, in the 75-year-olds, studies show about 1 in 3 people will have some signs of macular degeneration. So it's not surprising that in Last of the Summer Wine, 1 of 3 individuals were affected. And then if you go on to people in their 80s, by age 85, 1 in 2 people will have some signs of macular degeneration. So it really is an epidemic of visual loss, and as Helen's mentioned, is therefore very worthy of research funding, because it's such a major problem for the country. Now, not so long ago, when I considered myself a young eye doctor, it's actually about 20 years ago, there really wasn't anything you could do for macular degeneration. Patients came in and you said, terribly sorry, you've got this condition, and you discharged them because there was no treatment. Fast forward 20 years, and because of research, we now have some very effective and exciting treatments, particularly the treatments currently available for wet macular degeneration. So there is, over that time period, you can see evidence that research makes a difference and is helping to preserve vision. And we'd always love our research to move quicker, but I do see evidence, good evidence, that we're making progress. Now, one area that we think is important to understand macular degeneration is understanding the genes that, if they go wrong, lead to the development of macular degeneration. So our bodies are coded by DNA, and there are 3 billion pieces of information in our DNA in every cell in our body, and these bits of information are like the instruction manual for how to build a body. And by understanding what those instructions are, you can identify the pathways that lead to building a body and understand what's important when something goes wrong. And so there was a huge research project undertaken to decode this genetic information, and this was a Human Genome Project. And a few years ago, about 10 years ago, to decipher this code, this DNA code, in one individual cost about £20 million. To look at those 3 billion pieces of information. Now, a few weeks ago, I had a patient, a young man in his 20s in my clinic, who has a very specialized form of macular degeneration where the cone cells in the center of his eye don't work. And so he has difficulty with color vision, and he has reduced central vision. And the cone cells are the cells that are mostly in the center of the eye and the macula. But because of advances in deciphering the code, I was able to screen every single bit of his coding DNA for £800. Now, 10 years ago, that would have been £20 million. And I was able to identify the one change in 3 billion that causes his disease, which is a mutation in an enzyme called retinal guanylate cyclase. And that's important because it helps us understand exactly why he can't see, when his eye actually looks pretty normal, but I can tell him exactly why on a molecular level his vision's impaired. And hopefully eventually we'll be able to develop treatments to target that enzyme that's not working. And we can also give him genetic counselling and advice about risks to other family members. So it's anticipated that, and this technology currently takes a month or so to do that, which is really truly remarkable that you can look at that amount of information so quickly. But it's predicted that genetic sequencing will progress so that we'll be able to sequence an entire genome, an entire DNA code for an individual within 24 hours in the next 4 to 5 years at considerably reduced costs. And the costs, as I've said, have come way down, which makes it easier to push forward with research. Some insights from studying genetics. It used to be thought that to make a human required 100,000 genes. and 20,000 genes to make a banana. And we now know that it still makes 20,000 genes to make a banana, but it only takes 30,000 genes to make a human. So we're not quite as sophisticated as we thought we were. But related to macular degeneration, a major breakthrough has been by studying the genes of people with macular degeneration, the genes of people who don't have macular degeneration, and looking what's different. It's been identified that in many patients with macular degeneration, there is a fault in genes that code for proteins called the complement system. And so in about 60 to 70% of patients with macular degeneration, if we look at their DNA, we will find a fault in one of 3 or 4 genes that code for proteins called the complement system. Now, the complement system is a set of genes that make proteins that fight infections. There are complement proteins in your bloodstream, and complement proteins are made in your liver, and they're made inside your eye in these cells called the retinal pigment epithelium and also the choroid. And so what we now think, having started by looking at genes of people with macular degeneration, we now think that the complement system is overactive in patients with macular degeneration, and so there is a low-grade inflammation in people with macular degeneration that is causing damage to the cells in the back of their eyes. And so if we take a blood sample from someone with macular degeneration, we can measure increased complement activity. This process that's meant to fight bacteria is actually a little bit too active, and we think that's leading to damage over a lifetime in people's eyes with macular degeneration. And so these complement proteins can be found in these little yellowish deposits in the back of the eye called drusen, and so we now have a target for a new therapy for macular degeneration, and that is to inhibit the complement system. And so it's already been shown if you put drugs that inhibit the complement system into patients with wet macular degeneration, it will help treat it in the same way as these drugs such as Lucentis work. And it is hoped that it may also be helpful as a treatment for dry macular degeneration, and that sort of pertains to the question earlier, what can we do for dry macular degeneration? Well, we need better treatments, but one possible treatment might be complement inhibitors, and there are around 10 different complement inhibitors in early clinical trials now, some of them looking specifically at dry macular degeneration. We don't know yet whether it's going to work. We have to wait till the trials report, but it's an important new therapeutic target which came totally unexpectedly by studying the genes of people with macular degeneration. We have recently in Southampton looked at liver transplant patients, and the reason for that is because these complement proteins are made in the liver as well, and so by giving someone a liver transplant, you can change the complement proteins that they have in their body. And some very recent research that's come out of that is to identify that liver transplant patients actually have a higher rate of macular degeneration, which was totally unexpected and a novel finding, and also that you need to give complement inhibitor drugs inside the eye rather than in the peripheral bloodstream. So I think we're still going to have injections one way or another into the eye. The other thing you can do when you identify a gene that causes a problem is you can think about replacing that gene. And that's called gene therapy, and it's a simple concept. We can manipulate viruses, which are normally bad for us, but viruses are simple genetic organisms, and we can take out the proteins in the virus that cause disease, and we can use the virus as sometimes described as a parrot of a cell, and you can infect the back of an eye with a virus carrying a gene that you want to put into the eye. So the virus infects the cells in the back of the eye and delivers the gene you want it to deliver to restore function. So So, for example, there's a rare form of retinal degeneration called choroideremia, where there's a faulty gene, and because of this, your retina slowly atrophies. It slowly fades away. It's a little bit like dry macular degeneration. Up until now, nothing has been able to be done for that disease. But we've recently, working with colleagues in Oxford, been able to engineer a virus to carry a healthy copy of the choroideremia gene. That gene has now been placed under the retina of patients with choroideremia, and so for the first time in the world we have a potential treatment for this genetic disease. And there are other examples of other retinal diseases, particularly a condition called Leber's congenital amaurosis, where children, which again is previously a totally uncurable disease, where a virus has been used to put in a normal copy of the gene that's faulty in patients with that condition. And so children who were born blind are now riding their bicycles, and children as young now as 4 have been given gene therapy into their eyes to reverse this fault. And we have an advantage in the eye that when we're trying to deliver a treatment like gene therapy, because the eye is a small organ. You only need a small amount of genetic vector or virus to have a big effect. We also have a clear view of the back of the eye, and compared to colleagues who are, say, trying to do gene therapy for heart disease or muscle disease, we can see exactly where we're putting that genetic vector. And we have 2 eyes, so you can have one as a control to see what response you're getting in clinical trials. And so gene therapy throughout medicine has really been most successful, I would say, at the moment for eye diseases, and it has had the biggest proof of concept. Now, we're talking about the burden of disease with these repeated injections of antibodies against vascular endothelial growth factor, these Lucentis injections, and how people have to be repeatedly come up for injections. Well, it is possible to put genes that make proteins that inhibit new blood vessel growth into genetic vectors as well. And so you can give a single injection of a genetic vector making a protein that's similar to the proteins that are injected every month. And clinical trials have just started with this approach for macular degeneration. And so I was at a meeting in the United States just a week ago, where early results of this treatment for macular degeneration were described by one group of investigators. And basically, after these patients had 3 injections of Lucentis, they also had this genetic vector given at the same time, and they didn't require any further injections over a year rather than that first 3 injections, and got good visual results because there was a genetic vector, their eyes were making the protein needed to suppress new blood vessel growth inside their eyes, and they didn't need these repeated injections. So this is one example where innovative approaches, hopefully long-term, will give us long-term delivery of drugs or proteins to stop us having to repeatedly inject patients. And it's early days. It's what's called a phase 1 clinical trial, and there has to be several other stages of development to prove it's safe before it will be widely available. But it again shows the power of this technology of gene therapy. There's also a lot of excitement about something called stem cells. And our previous speaker mentioned how the eye can't repair itself. That if it's damaged, you develop scar tissue and your eye does not repair. And that's absolutely true, but it's not absolutely true in other animals, and particularly in reptiles. They have a remarkable capacity to regenerate tissue. So it's been known for 50 years that if you remove the retina from a salamander, it will regrow it. It has this remarkable ability to regenerate its retina. if you damage the optic nerve of a goldfish, and I don't recommend this, but if you do, it will regrow its optic nerve. And these are things that we can't do. And the reason that these animals, particularly reptiles and fish, can do this is because of they have a group of cells within their eyes called stem cells. And stem cells are cells that have the ability to be reprogrammed to any cell type. And what has recently been discovered actually is that we do have similar stem cells in our eyes and in our skin, but we have lost the ability to switch them on and to regenerate tissue in the way that other animals have. But we do have these cells in our eyes, and so we have retinal stem cells, and we can reprogram cells from our skin. We can take a skin biopsy, and using gene therapy or using specific proteins in a cell culture system, we can take skin cells and reprogram those skin cells to become these stem cells again. That's called induced pluripotent stem cells. And then get those cells to become retinal tissue, become retinal pigment epithelium, and grow these sorts of cells that we need to see in a culture disc. And that's truly remarkable. And indeed, about 2 years ago, scientists in Japan showed you could take skin cells and reprogram them to form almost an entire eye in a dish. By using a three-dimensional culture system, you can watch an eye develop before your eyes in a dish. So it's truly remarkable potential. Not yet proven in humans, and some of the challenges are, firstly, making cells that are safe, that won't cause an immune reaction in the eye, making cells that will make the connections with those nerves that go back to the brain. And that's the challenge, to get integration of those cells into our own eyes, which is something that reptiles and fish can do, and we need to understand better how to do that. Now, if you were a mouse, we could cure you. It has been shown that you can take stem cells in a mouse, which isn't that far away from a human, and a mouse that's blind, and get those stem cells to make those connections within the mouse retina so that the mouse can see again. So colleagues have put mice into swimming pools where there's a hidden platform and a visual cue where the platform is, and the mice that have had the stem cells put in can find the platform because of the visual cue and get there and get out of the water, which they don't like. But the point is, it shows they have functional vision, and that's very exciting because it's an important proof of principle that you can get integration of stem cells again. The challenge is in mice, you can take baby mice and get stem cells from them, and that's the type of cells that have been used to show this works. Now, we can't get baby humans and use those cells to create a therapy, and that obviously wouldn't be ethical. But the hope is, now that we've learned that you can take skin cells, reprogram them into the type of stem cells that we need, that we could take a patient's own cells and create the same sort of cells that have been shown to be useful in mice, and develop a therapy. And to that end, there are 3 or 4 clinical trials planned to start within the next year in humans using stem cells, and particularly to replace retinal pigment epithelium, because that's the easiest cell as yet to make from stem cells. And so we're actually got to the stage of human clinical trials. It takes a lot. I have to emphasize, a trial is not a treatment. It's an assessment of whether a treatment's safe and useful. but it's very exciting that we've gone from the eye's unrepairable to we're thinking about repairing human eyes in the next year. And so to that end, the Nobel Prize for Medicine last year was given to two scientists who have done some of the landmark studies on stem cells. And one of these scientists is a gentleman called Shinya Yamanaka. And he said, after he won the Nobel Prize, "We need to double check we don't see any severe side effects in patients after transfer. That's where we're spending most of our time. We are getting closer and closer. In some diseases like macular degeneration, it's almost ready to go." So he said that 6 months ago. And the clinical trial for macular degeneration, one clinical trial starting in Japan where he works in the next year. The other thing you can do with stem cells is you can take cells, as I say. Now, the problem we have with the eye is it's hard to get tissue to study. We can't go in and biopsy the retina of patients and to understand what's going on by looking at their tissue. We can't really go and say, can we have some of your retina? But what we can do with stem cells is we can take skin cells from a patient who's got macular degeneration. We can reprogram those cells from the skin into retinal cells, just like the cells in the back of their eye, and then we can study those cells in a dish. And that's a very exciting, very new thing. And I think that's going to help us to look at drugs that in a culture system that potentially could be helpful, again, for conditions like dry macular degeneration, because we now have a way to study the retina in a dish from patients genetically predisposed to the condition. And so we have again, not just for therapy, but for studying drugs, we will have a much more effective way to study the tissue we're interested in. The ARAD studies that you heard about, the vitamin studies, each study costs £25 million to do. We've got to have a more efficient way to study drugs rather than spending 10 years on one drug in patients. We've got to have a more efficient way to get the therapy. We can't spend £25 million every time we want to change a vitamin. And so this technology, I think, will help us to do that. So just going back to thinking about the anatomy of the eye, I said that the vascular layer at the back of the eye is called the choroid. And because of better imaging technology that we have, these OCT scans that I'm sure many of you have had if you've been treated for macular degeneration, we can now visualize the choroid much better than even a few years ago. And we realize that these vessels, these choroidal vessels, are affected in macular degeneration. And in dry macular degeneration, it seems that these blood vessels die. And so the choroid is becoming an area of interest for therapy. And currently we're looking at whether we could put stem cells back into the choroid, and there is some evidence that that might be possible. So stem cell therapy may be a way to regenerate blood vessels in the choroid for dry macular degeneration. I then mentioned above that there's a layer called Bruch's membrane, and as you develop macular degeneration, little holes develop in this, allowing the blood vessels to come through. And we and other colleagues are looking at ways to regenerate the Bruch's membrane with artificial polymers. So you can spin very fine fibers of the same sort of polymers that are used to make lenses after cataract surgery and are known to be safe in the eye. You can spin them in what are called nanofibers and recreate Bruch's membrane. And again, that's been shown to have potential to replace damaged tissue. And then I mentioned the retinal pigment epithelium as a layer above Bruch's membrane. And people have tried to inject retinal pigment epithelial cells back into the eye in the past, and it hasn't worked because the cells, instead of forming a nice flat sheet, have formed clumps. They've all clumped together rather than being a nice smooth carpet. But what we hope now to do, and has been developed, is to replace retinal pigment epithelial cells derived from stem cells on an artificial Bruch's membrane and surgically reimplant that. And again, that's getting very close to clinical trials. Above that layer is the photoreceptors, which, if the retinal pigment epithelium is damaged through macular degeneration, those cells die. Well, we're hoping by replenishing retinal pigment epithelium, we'll be able to keep photoreceptors to see better. Possibly with gene therapy, we can inject complement inhibitors that will prevent the damage that leads to damage to the retinal pigment epithelium and to the photoreceptors. But in patients who photoreceptors are dead or not working very well, if we can make a connection to the next bit of wiring, the ganglion cells, then we could bypass the problem. And so that's where artificial retinal implants are being developed. And there are currently 2 artificial retinal implants that are, one's been approved for use in the United States. One is very close to, is just becoming available in Europe. And these are essentially cameras that are placed underneath the retina, or one's placed underneath the retina and makes a connection with the ganglion cells. So light strikes the camera, it generates an electrical response, and that response is then carried with the cells that are still working back to the eye. Now they're still very crude, but the implant that's been made in the United Kingdom has 1,500 pixels. So that's like a very crude early computer screen. But patients who have no light perception have been shown that they can detect a knife and a fork on a plate. They can see the chin on their child's face. They can see the face of their child. And they can navigate through a room. They can see a doorway. And that's really remarkable in patients who have no perception of light vision. It's not, they can't read, and but it certainly is a significant breakthrough, and I'm every confidence that these devices will develop more quickly in the future. I just noted down what one of the people involved in making these devices said, a gentleman called Mr. Meck. He works for the American device. He said, while there are many academic centers working in retinal implants, getting the system to market took 14 years, $200 million, and a lot of intestinal fortitude. So at the moment, these cameras are just licensed for rare diseases such as retinitis pigmentosa, but I looked at the websites of both companies that are making these 2 devices, and they both said that their next goal is to use these devices for macular degeneration once the technology improves. So I think that's exciting as well. So we have approaches for every layer of the retina to try to stop or reverse the problems of macular degeneration. Regarding new lenses, there have been certain developments in telescopic lenses to try to magnify light that comes into the eye. So as I said, the macula is a tiny area in the center, and the way that magnifying glasses work is to project the image onto the more healthy peripheral tissue. And so you can, in a sense, put a magnifying glass inside the eye to create that magnification effect. I think it's early days yet to see how effective that is, and if you have good vision in one eye and you have a magnified image in the other eye, you may get double vision. So I think we need a lot more research to really see how much better they are than a magnifying glass, and currently they're very expensive and not available by paying large amounts of money privately. I'd like to see more clinical trials of this to really demonstrate how they compare to low vision aids before strongly recommending them. But I'm sure they are improving too, and the magnification's getting better. So just to sum up, Woody Allen said, "My brain, that's my second favorite organ." I'm sure he meant the eye was his first, and it's certainly mine because of the opportunities it gives us of developing cutting-edge new therapies. And really, eye research is at the forefront of some of these exciting developments in gene therapy and stem cell therapy. And finally, it's sometimes said that science is the pursuit of truth, but research is the pursuit of funding. And that is very true in the UK, and that's where the Macular Society is so important. It's always had as one of its founding virtues that it would support research into macular diseases, and not all patient groups do that. And I think it's very impressive that it's had that vision of supporting research, and it's been incredibly helpful to the research community that it has and helped drive this research forward. So thank you very much.